right, so just to help refresh where we were, last time we met on Tuesday, we summarized our skeletal muscle physiology and mechanics, and then we transitioned into our smooth muscle. We basically summarized where we left off on Tuesday is that there are two functional subcategories of smooth muscle. What are those two functional subcategories of smooth muscles? Single unit and multi-unit. We know that whether a smooth muscle operates as single or multi-unit is definitely going to be extrinsically controlled by the what? Either either multi-unit or single unit are extrinsically controlled by the the autonomic nervous system. We said the autonomic nervous system gives us involuntary control over things that we can't voluntarily control. So you can't tell your stomach to stop growling in the middle of class because it is controlled by the autonomic nervous system. You can't tell your bladder to stop contracting when you've got to go pee and you're sitting in class because it is controlled autonomically. You can't just do that. So they're definitely controlled extrinsically. We also, by the autonomic nervous system, we also know that, of course, these are parts, cells in your body. So sure, other things in the blood can influence it. We would say morally regulated. And certainly hormones. We know they can target the action of our smooth muscles like, say, prolactin, or oxytocin. I'm going to keep doing that. Oxytocin can target smooth muscle, for example. So hormones, nerves, and blood factors we know can control our smooth muscle. What we added is that some smooth muscles are also controlled how? What? Intrinsically. And the cells inside of the smooth muscle organs that give this intrinsic control are called pacemaker cells. And so on your handout, you had to pick the same picture from your textbook, and you had an autonomic nerve and some some smooth muscle cells. And it said just pick one. So in your picture of all the 8 options, just label one a pacemaker cell. It's the one that is going to just spontaneously do what? It's going to what? Depolarize to threshold and generate an action potential. It is connected by what? Gap junctions to other cells which work as contractile cells. The other cells, their job is to contract. Pacemaker cells generate action potentials intrinsically, and because they're connected to contractile cells by gap junctions, the contractile cells will correspondingly contract. The mechanics of which we have not talked about yet, but you had to kind of get ahead, answer that last question in your handout about the excitation contraction coupling in your smooth muscle. So you actually had to look ahead to answer that. And what you should have taken away is that it's very much different in the smooth muscle than it is in the skeletal muscle. So before we get into the contraction inside of a skeletal muscle, let's go through and look at the pacemaker potentials that are in the pacemaker cells. And so the pacemaker cells, we said, spontaneously depolarize to threshold regularly at a steady pace. And that's why they're called pacemakers. If you turn to figure 1237, it'll help illustrate this change in polarity within the within the pacemaker cells. You'll see that they'll continue to grade up to a threshold and then depolarize and generate an action potential. And then they'll grade back up just continuously, just regularly, intrinsically. They're going to do this. So what you can add about your pacemaker cells is that they're leaky. They're leaky. They're continuously leaky to what type? Ions. Ions that have sodium as an example. They're also leaky to calcium, which have what type of charge? Positive. And so we leak positively charged ions into a cell. It is going to depolarize up to threshold in the pacemaker cell. what questions do you have about using the word pacemaker cell in some smooth muscles? The contractile cells do this? They don't, because they're not as leaky. They will respond, they can generate action potentials, but not just randomly, continuously, whenever. Only when triggered by the pacemaker, or even by nerves or something in the blood or hormones. They could be intrinsically regulated, yes, but they could also be extrinsically regulated. All right, so again, what questions do you have about how the pacemaker cells are considered to be leaky and continuously depolarize to threshold? Or maybe how they're physically connected to the rest of the muscle cells by gap junctions.
So every time they depolarize the threshold, they make an action potential through all of them? Or?
Yes. It'll start in the pacemaker cell, and then it'll just spread through the rest of the muscle cells.
Is there any way for it not to? How would it mess up?
It could mess up. You could turn it off by, so the autonomic nervous system could override it and say, maybe blocking calcium channels or blocking sodium channels, for example. So the pace could be slowed down by the nervous system. It can even be sped up by the nervous system. So the extrinsic control could certainly influence it. But because they're leaky, they're going to just normally do this. But you're correct in that it can be influenced extrinsically. And so some neurotransmitters would tell to slow down. Like say, if this is in your stomach, the sympathetic nervous system, so you're running, you want your smooth muscles to be real active in your stomach? No. We want them to relax. And so epinephrine would tell those smooth muscle cells to slow down, not depolarize so quickly. Good question. Additional questions? So what type of smooth muscle has a pacemaker? Single unit. Because all these physical cells that are connected together, even though there may be thousands of these cells, they act as a what? As a single unit. Furthermore, what we mentioned a moment ago is when a smooth muscle cell undergoes this excitation, it's very much different than it is in the skeletal muscle. So it took us a couple of weeks to talk about how the skeletal muscle does its excitation contraction coupling. It's quite different in the smooth muscle. There's not a lot of great detail on structure of the smooth muscle in your textbook because this is really still being studied. So there's not a lot of definitive answers out there. And so I pulled an article from 2010 to help illustrate that we're still learning this, how the smooth muscles actually really work and the difference in structure. And so what we're going to do is talk about the excitation contraction coupling. There's a big spot in your guided notes, or you can check your work in your handout. So we're looking at what type of cells. If we're looking at excitation contraction coupling, we're looking at which type of cells in your smooth muscle? Which ones? The contractile cells. So now we're transitioning to the working machinery of the smooth muscle. So in your contractile cells, the series of events that lead up to contraction are very much different. The first thing is these still have to depolarize to what? Threshold. The first difference is that the depolarization to threshold and source of calcium is very much different. So this is the first difference, is when a contractile cell grades up to threshold, voltage-gated calcium channels at the cell membrane open. And so calcium will flow where? Into the cytoplasm. So our calcium comes from where? It goes into the cytoplasm. It came from the outside, the extracellular. It comes from originally from the extracellular fluid, gates open, and then it goes into the cytosol. So that's different. Where did skeletal muscle calcium come from? From the sarcoplasmic reticulum. From the smooth muscle, calcium floods in from the extracellular fluid first. Then secondarily, we can see a little bit of calcium come from where? There are no T-tubules in our smooth muscle. It will come secondarily from the sarcoplasmic reticulum. So first, calcium comes in from the extracellular fluid. That causes the release of calcium from the SR. First comes from the extracellular fluid. That leads to calcium from the SR, the sarcoplasmic reticulum. So we've got two sources of calcium here in the smooth muscle. But what's notable is the bulk of it comes first from the extracellular fluid. Then you can see some coming from the SR. But a common event is still we need cytoplasmic calcium. So calcium is still going to be our messenger for contraction in the smooth muscle. That's about as where the commonality ends. Calcium is going to have a different mechanism in the smooth muscle than it did in the skeletal muscle. And there's something just flat wrong in your textbook, and I want to address it. And it may be because of the data is still just kind of coming in, or they just had a flat out error. I don't know what happened. But in your textbook, you may notice that it said the smooth muscles have troponin but not tropomyosin. Anybody read that?
Right.
Completely backwards. No troponin and something like tropomyosin. No troponin. Smooth muscles do not have troponin, and they have something that works like tropomyosin. Again, your smooth muscles lack troponin, and they have something like tropomyosin in them. So clearly, calcium cannot bind to. Can't bind to troponin, so there's no troponin there. Because what it tells us is that it binds to something completely different. Are we okay to move on now? So calcium will come after a depolarization. Calcium will flood into a smooth muscle cell first from the extracellular fluid, and then secondarily from the SR. It will then bind to something called calmodulin. C-A-L-M-O-D-U-L-I-N. Cal, C-A-L, mod, M-O-D, ulin, U-L-I-N. Calmodulin. This is a protein that modulates. It binds to calcium. Calcium binds to calmodulin. We're going to basically see just kind of a signaling pathway. Does anybody know what, if you did your work, what does calmodulin, this calcium calmodulin complex bind? What does it bind? What does the calcium calmodulin complex bind? Myosin what? Myosin light chain. What's the kinase? All right, so something called myosin light chain kinase. You can just call it myosin kinase. So not myosin, but something called myosin kinase. So let's make sense of this word. Kinase, how do we spell that? K-I-N-A-S-E. Kinase, it is an enzyme. myosin binds to an enzyme called myosin kinase. What does that enzyme do? Kinases, we studied earlier in the semester,
Determine.
catalyze phosphorylation. They transfer a phosphate group. They transfer functionally. When we give something a phosphate, we give it energy. These things phosphorylate. What's the first word? Myosin. So I'm going to back up. Calcium calmodulin complex activates an enzyme called myosin. The light chain kinase, the light chain part is important, but I'm just going to, so you get confused, just call it myosin kinase. The calcium calmodulin complex can now bind to what? We just said? Myosin kinase, which is an enzyme that phosphorylates myosin. Well, that's good. Because myosin is what type of protein? Cell biology? What type of protein is myosin? What can we call it? It does work with the contractile protein. The word I was fishing for is motor protein, but I don't know that we use that. She saved you. It's a motor protein. It is used for contraction, which is good because it's a muscle and it needs to contract. Okay. All right, so I'm going to go back and just repeat so you can check your notes. When a contractile smooth muscle cell gets excited, it's going to let calcium in from the extracellular fluid and then also from the SR. Calcium can now bind to calmodulin. That can activate myosin kinase. When activated, phosphorylates myosin. Gives myosin, this contractile motor protein, the ability to contract, do its power stroke. So the myosin, same thing, is still going to have to cross bridge over and bind with actin and power stroke it. Those are the excitation contracting events in a smooth muscle. So turn your attention to this fairly recent article from 2010. This is showing a smooth muscle membrane voltage-gated calcium channel in the membrane, letting calcium in, which then lets calcium on the SR out in the cytosol. Calcium binds to calmodulin that activates our myosin kinase. It can then phosphorylate myosin. Give it some energy to do some work, such as contraction. This will be a look at the third panel. Just zooming into the little myosin cross bridges, the phosphorylation has taken place, and that powers the contraction. The thin filaments are not shown in this particular image. I've got another image I'll show you in just a second. So
Does that mean just one phosphate?
yes, per head. Each myosin molecule gets its own phosphate group. Yes?
What were the steps after the calcium calmodulin complex activates myosin kinase? What is the step after that?
Myosin kinase then transfers a phosphate over to myosin, which gives it energy to work, to contract, to power stroke. Yes?
Do these do the walking or the tugging thing?
They do pretty much identical to what you saw in the skeletal muscle. And so let me go ahead and pull that image up since that question is here. Question is, how do they work? And so, same concept, so still have a thick filament, and so they still do the walking and the tugging. So what happens is, instead of a sarcomere arrangement in your smooth muscle, your thin and thick filaments are spread out kind of diagonally. And so you get it pulled together, short and broad and all kinds of stuff. But the concept is the same, you're correct, and that the cross bridges walk, motor, and when they do that, the thin filaments are brought towards the middle. You would not use the word inline, nor would you use the word sarcomere, because we're in a smooth muscle, not in a skeletal muscle. But they are brought towards the center of this contractile unit. I don't know what the right word is. You can't use the word sarcomere though. It's not banded like that.
What are the things going diagonally? The blue lines?
The blue lines are intermediate filaments that serve for a point of attachment. In the red and the yellow are representing the thin and the thick filaments.
And then they pull the intermediate filaments, which makes the whole thing go in and out.
Yes. And so the method of contraction is different physically in a smooth muscle than it is in a skeletal muscle, due to the way that the way that you noted how these are arranged differently. In a skeletal muscle, everything's just parallel one another. Here in the smooth muscle, it's splayed and diagonal. And so an entire smooth muscle organ won't just shorten like your skeletal muscle organ. So someone had a question about this in yesterday's class too, and a very over exaggerated example is, for example, when your stomach digests food, you don't see the whole digestive system shorten from the esophagus to the colon. You're like, it's not, you're like shrinking in pain because those smooth muscles are shortening long-wise. They're shortening long and spreading, and these are lining hollow organs. And so some can constrict a little bit. It's just, someone gave an analogy of if you've ever seen a snake eat its food. I can see the little smooth muscle contractions kind of just move through the little animal, kind of basically what happens inside of your smooth muscle. So some of the whole cell, the whole organ doesn't just shorten like a skeletal muscle cell. It may shorten this way, it may shorten this way. The whole thing will just change its state. But these cells are arranged in sheets, and so when one pulls this way, they may all just, it's described as sheets. You think of it like a tube and cells. I'm having trouble with the visual to help answer this. Can somebody help me out with a visual?
So it's not like the little kid ball where it's like all spread out and you can pull one and then it goes in. There's like sticking everything. You can pull one and it gets round.
Correct.
It's not like that.
Correct. So I think what are those? The sphere, geez.
I don't remember.
You know what she's talking about? The little compacted? It's not like that. It would be like that if it could maybe shorten and flatten out. That's a good analogy. So if you could take those little, we're talking about the little, what are they called? You know what I'm talking about? We're doing this. Someone's gonna listen to the video and go, I have no idea what's going on. Like geodesic something balls or whatever. That can constrict, if you will. And then, so if you could take that thing and then its shortened state, squish it and all those, so it will become like a frisbee. And so say going to a circle, it would go to a frisbee. That's kind of how that's what I'm trying to describe your smooth muscles and what they would do. So it'd be like that ball. They shorten it and spread it out. And it shortens. Question? Were you going to give me the name?
No, I was just gonna, because I looked at a lot of pictures to try to find that, and so I saw some pictures where it looked like a quilt almost, and like the strings of the quilt had shrunk, but not the quilt, so then it was just like, and then the bubbles out the sides, you know?
Yeah.
So like,
Yes, like what I was flattened. And the reason there's a lot of not great images, again, still working on it. Still. We have the idea about the basics. We don't have exactly what's going on. So did that answer your question at all about how the thick filaments motor along the thin filaments? So I want to go back to the presentation and just review those excitation, contraction, coupling events. Those are summarized for you in figure 1235. So we're just going to keep repeating like we've been doing for the last 20 minutes. when we look at excitation, we said calcium first comes from the extracellular fluid. That triggers the release of calcium from the SR, still goes into the cytosol. That will bind to calmodulin. Calcium activated calmodulin can bind to myosin kinase, okay, shown here in the green. That will go on to phosphorylate myosin, and the muscle will contract. So we still need calcium, we still need ATP, but the actions are in different locations because the muscle cells are built differently. Yes?
So the myosin kinase, it takes phosphate to the myosin?
Yes.
Okay.
Yes, it transfers a phosphate from ATP over to the myosin, yes, to the myosin.
Is there some sort of receptor on the calcium channel? And on the ER?
Those are usually voltage gated channels, but you can have some ligand gated calcium channels to initiate muscle contraction. Specifically, if you're looking at your multi-unit smooth muscles, which rely exclusively upon extrinsic control.
So the calcium coming in, the voltage is what makes the calcium release from the ER?
Yes. The question is, what causes the SR, or the ER, whatever you want to call it, from what causes it to release its calcium, and it is the voltage change from that first source of calcium. We cause it to depolarize, and those voltage gated channels open up. So these are voltage gated calcium channels. That's the ER, smooth ER. So again, this is summarized in figure 1235 and the corresponding couple of paragraphs around it. Before we move on, what questions do you have over this excitation? If the smooth muscle needs to relax, what should happen to it? You could hyperpolarize. What else could you do? First, you need to unbind the myosin, detach the myosin, detach it from the actin, and even do what? You need to unphosphorylate, or we've been using the word dephosphorylated. If the myosin is dephosphorylated, it doesn't have any, phosphate doesn't have any energy, so it can't contract. And so we need to phosphorylate it, and the way it's phosphorylated is by activating myosin kinase, which has to first be activated by calcium calmodulin, which has to be first activated by this, which comes in from the cell from the extracellular fluid. And then we went ahead and reviewed the anatomy of the thin and thick filaments is different between the smooth muscle and the skeletal muscle. Here's another image from 2008. May help support kind of how the muscle changes state in the relaxed state in the top panel and the contracted state in the bottom panel. This will help maybe answer the question you had: where do these thin and thick filaments go? And they shorten, and we have these kind of they're called dense bodies in the middle, and they're points of attachment. Instead of just having them pointing at attaching into ends like you have the skeletal muscle, those various branching points in the middle of the muscle cell gives that splaying, spreading type of, not just shortening but also spreading out type of contraction. Does that bring up any questions? Yes.
Not about that, but in the picture it shows the endoplasmic reticulum, but you said when we talk about muscles, it's the sarcoplasmic reticulum?
Yes.
So in this picture, that should be called the sarcoplasmic reticulum.
Yes, it does. The sarcoplasmic reticulum is the endoplasmic reticulum. It is more specifically the smooth ER. Technically, you wouldn't want to call it the sarco in the smooth muscle, but I'm okay with you using it. Because sarco talks about flesh, and that means your skeletal muscles. This is not skeletal muscle, this is smooth muscle, and so they could just say smooth ER, they just have ER. I'm okay with you calling it the sarcoplasmic reticulum. It's, either way, if you don't want to go with that short name, you can just call it what? And more specifically, you can call it the smooth ER, smooth endoplasmic reticulum.
So for test purposes, you don't care what I put.
Correct. Correct. Because by all technical definitions, if you go on and become a, I don't know what the name, myologist, I'm not sure, muscle, person, study, myologist is probably a word, I don't know. Then you might just say, well, that's smooth ER, sarcoplasmic reticulum for skeletal muscles, and I'm into smooth muscle, that offends me, so I'm gonna call it smooth ER. So someone might get hung up over it, I don't, on that one. But if you wanted to call it the rough ER, would I get hung up on that? Yes, because it's not the rough ER, it's the smooth ER. If you wanted to call it the Golgi, would I get hung up on that? Yes. If you wanted to call it the cytosol, would I get hung up on that? Yes. Does that answer your question?
So would it work inversely? Could you call the smooth ER in the muscular?
You could say smooth ER in the muscle,
Okay.
skeletal muscle, cardiac muscle, smooth muscle. You sure could.
Thank you.
As long as you prefix it with the smooth part. And then figure 1236 reiterates what you saw in your handout that you're going to turn in just a little bit, helping illustrate that each your multi-unit and your single unit smooth muscle are extrinsically controlled by the autonomic nervous system. However, we see that the multi-unit is more dependent, we say more innervated by the autonomic nervous system, because these cells are all structurally independent, exclusively rely upon that extrinsic control. By contrast, your single unit smooth muscle, still extrinsically controlled, but less dependent upon it, because it also has cells that act as pacemakers. And again, I don't care which one you label. Of the 8, just label 1 as a pacemaker cell. And the bulk of these are connected by gap junctions. And so the bulk of them would then be contractile cells. So if you want to, you could go through and just label, right, or draw the cross-linking of the thin and thick filaments to iterate that those are the ones that contract. You lack those in whatever you label as the pacemaker cell.
So the white part is the contraction cells, and the orange we can say pacemaker.
What white?
Inside the full protein.
In here?
Uh-huh.
So this is one cell. 1, 2, 3, 4, 5, 6, 7, 8 cells. I can't say which one is which because it's a very just simplistic image. Because I'm telling you, just pick one. Let's say this is our pacemaker cell. You would label that as your pacemaker cell. And then the rest would be contractile cells.
And is it like 1 to 7 for the ratio?
That's not the ratio. It may be 1 to hundreds, or it may be 1 to thousands.
But it is like 1 to a lot.
Yes.
Okay.
You would have one pacemaker. You would not have a 1-to-1 ratio. Because then it would just be a mess. It'd be like having to send out, like, if the United States had 50 presidents try to control the one nation. It'd be like having that, right? It just doesn't work. You have one president. You have one set of cells that work as the pacemaker.
Are you recording? It's not showing you recording.
No, I thought I was. And then a pacemaker is not going to be just a single cell either. A pacemaker is going to be several cells, which is good because so they would work together as also a unit. So if we go back to the president example, so the president has his cabinet. So the president and the cabinet help run the United States, or you could say all of the legislative branches, right? So they all work together, right? So it's not just one cell, which is good. So if you have a damage and you hurt that cell, one cell in the pacemaker, you still have controlling ability. But the proportion is still very few pacemaker cells, and the bulk in the contractile cells, because smooth muscle's job is really to do what? Muscle cells' job is to contract. Okay, so we want the bulk of these things to be contractile cells.
Like, are all the cells in like your uterus or your bladder, are they all connected through a gap junction?
Yes. When looking at single unit organs only, not in multi-unit smooth muscle cells.
Are there organs where there's multi-unit and single unit?
Not that I can think of. That control that organ. On Tuesday, we gave some examples of multi-unit, didn't we? Where you would find them?
Blood vessels and
Yeah, surrounding the capillaries, when it's going respiratory passageways. So you wouldn't have any single unit in those.
You said the multi-units and, smooth muscles are dependent on just the varicosity or viscosity.
Of the autonomic nervous system. And in the other extrinsic contracts, so not just the nervous system, hormones and stuff in the blood, humorally, hormonally, and neurally, but not intrinsically. Yes.
Humorally, that's just the stuff in the blood.
Like calcium, like glucose, like sodium, whatever. We're going to very briefly talk about cardiac muscle now, and then we're going to talk about it in better detail in chapter 13. And we're going to compare cardiac muscle to our other tissues. We're going to find that it's kind of a mixture between the different muscle tissues, but it's still muscle, so it's designed to, the bulk of it is designed to contract. Anatomically, if we compare cardiac muscle to skeletal muscle and that of smooth muscle, we're going to go back to more structurally what we're familiar with, and that the cardiac muscle is like skeletal muscle in that it's striated. So if you look at it under a microscope, you're going to see the light, dark banding pattern. If you see that light, dark striation, that tells you that the striations are arranged in sarcomeres, and the sarcomeres again have troponin and tropomyosin. So anatomically, it's built to contract just like what? Skeletal muscle. Anatomically, though, it's also like smooth muscle in that it works like a single unit smooth muscle. There's a set of cardiac cells that function as a pacemaker, which do what? Generate action potentials spontaneously, regularly. Furthermore, the source of source of calcium comes from our extracellular fluid, which is like our smooth muscle.
Does it also come from the endoplasm?
Yes, it then would secondarily come from the endoplasm, which is smoothie up. Furthermore, our pacemaker cell is connected to our contractile cells by way of gap junctions, which does what? Yeah, allows the diffusion of ions from cell to cell or conduction of action potential. Not only is it intrinsically controlled by pacemakers, it's also controlled autonomically, extrinsically by the involuntary nervous system. So it's a meld. The physical events of contraction in the cardiac muscle is going to be like what? Like the skeletal muscle. So calcium, even though it came from the extracellular fluid, is going to bind to troponin. It's going to be like the skeletal muscle. So calcium binds to troponin, pulls back, tropomyosin. Myosin binding sites are exposed, so myosin can cross over and bind. We now need what? ATP to power it. Calcium was there, right? And then we need ATP to power it. Muscle relaxes, so say a chamber goes into diastole, calcium goes back where? Extracellular fluid and maybe a little to the, so the extracellular fluid or the smooth ER. Alright, so let's go ahead and transition into the cardiac muscle, which is chapter 13. We may not get all the way through chapter 13, so wherever we get today is where our exam material will culminate. So if we go ahead and review the chapter 13 objectives, we're going to talk about which components comprise the cardiovascular system. We're going to introduce the cardiovascular system today with chapter 13. And then we'll come back next week, we'll continue, right? So the cardiovascular system is going to span the third and the fourth units in this course. We're going to look at major structures of the heart, which we kind of implied in lab last week. We're going to review how blood flows through the heart. I'm not sure that we're going to, we'll hopefully get at least to how the action potentials are carried through that intrinsic conduction system. So we're going to talk about SA node, AV node, etc. And then we may not get to point 5, cardiac cycle. We may not get to the ECG. We'll see how that goes. And at some point we'll link the electrocardiogram events with the physical events of the heart, which are called the cardiac cycle. And then we'll differentiate intrinsic and extrinsic control of the heart, which we have an idea about. Intrinsic comes from the intrinsic control comes from the pacemakers, and then the extrinsic control is going to predominantly come from the ANS, the autonomic nervous system. And then we're going to look explicitly where the autonomic nervous system actually acts on the heart, and introduce some terms called afterload, preload, and then contractility, as well as their influences on cardiac output, which we introduced in lab. Okay, so we probably won't get all the way through there, but we'll do our darndest. What we're going to first do is just talk about the 3 components of the cardiovascular system. The 3 major features of this system include the heart, which we're going to cover here in chapter 13. What does the heart do? Pumps blood. The reason it pumps blood is because it establishes a blood pressure gradient. The heart is going to establish a blood pressure gradient. And that's what allows it to pump and propel blood. That's what we're covering here in chapter 13. Chapter 14 is going to talk about the anatomy of the blood vessels and the pressure within them. So what do our blood vessels do? They transport and distribute the blood to the different parts of the body. We'll review some of their major physiology in chapter 14. Chapter 15, we're going to turn our attention to the 3rd component of the cardiovascular system, which is the blood. And we're going to focus predominantly on red blood cells and the plasma. We'll talk about that when we get into chapter 15. What does our blood contribute to the system? Gives us oxygen distribution. What else? Nutrient distribution. What else? Hormone distribution. Carbon dioxide distribution. What else? White blood cells heat. Very good. Fluids. Diet. So blood is more than oxygen. So we'll talk about that in chapter 15. We're going to now just turn our attention to the heart, because it is a muscle organ, and we have an idea about muscles, so we're going to continue that. The heart is located in the thoracic cavity. It's tucked neatly between your 2 lungs. It is an asymmetric organ, so it doesn't look like a mirrored image. So structure is different between the 2 sides, so the 2 sides might function a little bit different. And let's look at the heart in a little bit better detail. So let's label the heart. This is figure 13.1 in your textbook, and you have the same thing in your guided notes. Let's just label a few things. The heart has how many chambers? The human heart has 4 chambers. The chambers on the top are called atria. So Atria is plural, atrium is singular, so you would have the right atrium and the left atrium. So label this where my hand is on the right side of the heart, label that the right atrium. The smaller one on the opposite side is the left atrium. This thin layer right here, what is that? Yes, it's the atrium, but what's making it up? Muscle. What kind of muscle? Cardiac muscle. We are in the heart. So cardiac muscle is in the heart. So the atria are muscles. They make chambers. The other two chambers are called ventricles. They are also made up of cardiac muscle. The ventricle where my hand is here is called the right ventricle, and this is the left ventricle. We studied in class, or in lab, that each of the chambers can either be in a phase of systole, meaning that they contract and push blood, or they can be in a phase of diastole, where they're relaxing and filling. The right atrium, right ventricle, left atrium, left ventricle, cardiac tissue, or you could say myocardium, makes up the bulk of the heart. Separating the chambers are things called what? Valves. The ones between the atria and the ventricles are called what kind? Atrioventricular valves. Okay, so you can label that an AV, atrioventricular valve.
So on the test, is it gonna be AV? Cause then can't you call it a tricuspid?
You could call them a tri or a bicuspid. The tricuspid separates between the 1, 2, 3. Where do you find the tricuspid?
Right atrium.
Between the right atrium and the right ventricle. I tend to just go with the AV valve because that's, to me, it's the valve between the atrium and the ventricle. That's easy. I don't have to think so much on that one. However, there is an AV valve between the left atrium and ventricle. You would call that the bicuspid valve. What do our valves do? Prevent blood from going backwards, prevent backflow of blood. They are connective, dense connective tissue. They are not what? They're not muscles. They do not contract. They're pushed and pulled when pressure changes. So they're not muscles. There's a muscular mass between the ventricles. You might know what it's called. It's called the septum. You could call it the INTER. That means interventricular septum. The septum between the ventricles. And this is what keeps the blood in the correct chamber. Keeps it from mixing from the left and the right side. There's also valves between the ventricles and the respective circuit. What are these referred to as? You call them the pulmonary valve. Okay, the pulmonary valve separates which chamber? The right ventricle from which circuit? Pulmonary circuit. The pulmonary valve is between the right ventricle and the pulmonary circuit, shown here in this blue. So label this pulmonary artery.
Could you call that the semi-lunar valve too?
Yes, they are called the semi-lunar because they look like little moons, semi-moons, crescent shaped. There is a similar semi-lunar valve, between the left ventricle in which circuit? Systemic circuit. So what vessel is that? Aorta. So you could call this the aortic valve. What is the name of the blood vessel that takes blood to the right atrium? What are these vessels called? Vena cava cava, whatever. Vena cava, whatever, however you want to enunciate it. VENA CAVAE, vena cava. Are the vessels, and someone heard it correctly, there's a superior and inferior, but they drain into the right atrium. The SA node, where is it located? The top of the right atrium. So why don't you just draw, it's right around in here. So why don't you just draw darken an area of the right atrium where that superior vena cava drains, and just darken it, and that's where an area about where you would find the SA node, or the pacemaker cells. Alright, so let's go ahead and turn our attention to the two blood circuits. What were the two blood circuits? We have the pulmonary that goes to the lungs, and we have the systemic. Blood is propelled to each the pulmonary and the systemic simultaneous. So when the ventricles contract, blood is not only going to the systemic circuit, it's also going to the pulmonary circuit at the same time. We can look at how blood flows to the heart. We introduced it in lab in the handout, but we didn't talk about it, or in the lab last week, we didn't talk about it in all labs. So let's just go ahead and review how the heart, if you're to pick a single red blood cell and just kind of trace its way through the body. If we look at blood, say coming from the systemic circuit, it's already dropped off oxygen, picked up some carbon dioxide, maybe picked up some hormones, maybe nutrients, depending on where it's at. It's going to return to which side of the heart? Left or right? Which side? It goes to the right side of the heart. If it's coming from the systemic circuit, blood is going to enter into the right side of the heart via those vena cava. Those vena cava drain into which chamber? The right atrium. After the right atrium goes into systole, it pushes blood across the tricuspid valve and collects into the, or you can say AV valve, and collects into the right ventricle. From the right ventricle, the ventricle could go into systole. The AV valve will do what? As the ventricle goes into systole, the AV valves will close, and then hopefully the semilunar valves will open, and blood is pushed to which circuit? Pulmonary circuit. At the pulmonary circuit, the blood is going to unload which gas? It's going to unload carbon dioxide out, and we'll talk about those mechanics in chapters 17 and 18, or like 16, 17, somewhere around there. And we're going to load up oxygen. That newly refreshed blood will exit the pulmonary circuit and enter which side of the heart? The left atrium. The left atria goes into systole, pushes blood into the left ventricle. When the left ventricle goes into systole, that AV valve will close, semilunar valve will open, and blood enters into the aorta, or you could say systemic circuit, where it's going to now drop off which respiratory gas? It's going to drop off oxygen and pick up carbon dioxide. And whatever else is dropping off and picking up or whatever. What questions do you have over how blood flows through the heart?
Do the atria have valves where the superior and inferior veins come in?
Not where they dump in, but there are valves present in the veins throughout the body system. As far as though there's no physical valve here, because the blood pressure is super low, we don't need anything else hindering it. So it's just like, come on in, we need to fill back. There's not another set of valves. If there were, we'd need really another heart pump to squeeze that blood back into the right atrium.
Is the superior and the inferior, is it just like blood from the top and blood from the bottom?
Yeah, so the superior drains from the head neck region, and the inferior drains from the trunk of the body into the heart.
So like when the atrium contracts, can some of the blood go back out?
It could back out a little bit, but there's valves further back, and so it's not going to be significant. It's not going to go back very far. I don't know anatomically where the next valve, I guess first valve is, but they're present, so it won't back. If it does, it's not going to be significant. Good observation. And just to remind you, it's going to both circuits at the same time, but I can't double talk, since they got to go in sequence like that. Furthermore, the heart has its own systemic circuit, which makes sense if you've ever heard somebody had a coronary artery failure or deficit or whatever. The heart has its own systemic circuit. So the blood flowing through the heart is different than the blood flowing to serve the heart. Okay? If the systemic circuit to the heart is cut off, the heart is going to do what? Die. So we have a heart attack, and it's not going to work right, just like any other tissue in the body. The next image is figure 13.7. Oh, sorry, got ahead of myself. Excuse me, figure 13.6 is illustrating that the ventricles are, even though they function the same, are not built identical or symmetrical to one another. They use the word wall to describe, or I guess wall is an accepted term to describe the muscle of the ventricles. And if you look at the musculature of the right ventricle to that of the left and even the septum, you can see some really profound differences. The left ventricle is what? It's thicker when compared to the right. Because they need to propel the same stroke volume to each circuit, but what? Why do we see this difference in musculature? That's true. I don't remember what else. The systemic circuit has miles and miles, hundreds of, probably not, I don't remember the value, but it's like at least hundreds of miles of blood vessels in your systemic circuit, contrasted to the vessels that go to the pulmonary circuit. And so even though we still need to generate pressure to push blood to the pulmonary circuit, we don't need as much pressure. And so in chapter 14, we're going to learn that the more vessels there are, the more resistance there is to the flow of blood. And so your heart needs to generate a lot more pressure to push it out into which circuit? Systemic circuit than it does to the pulmonary. But it still needs musculature, just not as much. And so it's just an anatomical distance. It's greater in the systemic circuit, so we need more musculature to push that same volume, not only to it, but also from it. We need to get it back to the heart. Not only when we get it out of the heart, you need to have enough force to get it back to the heart. So that's why the left ventricle is so much thicker than that of the right.
Does the pulmonary circuit have valves throughout it?
I don't know about the, I would presume they're, I don't know if they're there in the, I don't know. Say that 3 times, but I'm gonna guess not. But again, I don't know about.
We'll get into that one.
Yes, but we really look at the systemic circuit. I'm not sure about the pulmonary circuit. Has anybody had anatomy? More recently than I have? That was so last century. Okay,